Within glucose metabolism, the initial process is to break down glucose via glycolysis to yield two pyruvate molecules. Now, those two pyruvate molecules must be converted into acetyl-CoA in order to undergo further oxidation via the TCA cycle. Now, once we get this pyruvate from our glycolysis, this pyruvate will be converted into acetyl-CoA with the help of the enzyme pyruvate dehydrogenase complex. And now this complex has three subunits, the E1, E2, and E3 subunit, but we're not going to go into that much detail in this video. But first, what we see is that TPP is involved whenever we have some sort of a carbon-carbon bond breakage. We'll see TPP there. So in this case, we see that we're cleaving this bond over here and we're going to be releasing CO2. So this is the part where TPP is going to be involved. Then we get this 2-carbon fragment intermediate, which is going to be transported via the lipoate, where our coenzyme A will then bind. Then once the coenzyme A binds to this 2-carbon fragment, acetyl-CoA is released as a product. And the electrons that are released in this process are picked up by FAD, which will pass it on to NAD+. Now, we'll go over the mechanism details in another video, but for now, all we need to know is that pyruvate is converted into acetyl-CoA and carbon dioxide is lost in the process. Now, this acetyl-CoA will now be fed into the TCA cycle. Now, over here, we can see that we are adding in our acetyl-CoA, which is a 2-carbon molecule. So this is our acetyl-CoA, CH3, then we've got this C double bond oxygen and that coenzyme A group. And now we're feeding it in, and we see that it's combining with this C4 compound molecule. So the C4 molecule is oxaloacetate. So oxaloacetate, it's actually a part of the TCA cycle. Like over here, we can see the cycle. Oxaloacetate is a 4-carbon compound that is a part of TCA. Now over here, we can see that this is the structure. So essentially what's happening is that oxaloacetate and acetyl-CoA, they're combining, okay? So they're combining to form citrate. So see, we get a 6-carbon molecule. So we had C2, C4, and now we have C6. And what happened is that they just bound to one another. And now there's a specific mechanism for this process. It's the citrate synthase mechanism, and that will be discussed in a different video. But all we need to know for now is that these two combine and we get citrate, and the enzyme that is catalyzing this reaction is citrate synthase. Now, once we have citrate, it undergoes an isomerization reaction to yield isocitrate. Okay? So we undergo this isomerization via an enzyme known as iconitase, and then we get isocitrate. So keep in mind that it's still a 6-carbon molecule. We've just switched where our OH group was. So we see OH was over here on this carbon, but we've switched it to the carbon below. Now, the next step, it's very important because we're releasing carbon dioxide. And if we're releasing carbon dioxide, that means we're losing one of our carbons. So now we're going from C6 to C4. So which carbon did we lose? We lost this carbon group over here, and this was released as carbon dioxide, and we don't see that anymore here. Now, the enzyme catalyzing this step is known as isocitrate dehydrogenase. Isocitrate dehydrogenase. Now, whenever we have a hydrogenase enzyme, that means we will be producing NADH. So we can see over here, NAD plus becomes reduced to NADH because we have a dehydrogenase enzyme. Now next we have a C5 compound known as alpha ketoglutarate, and we see that over here. Alpha ketoglutarate is also going to release CO2 as it produces the product succinyl-CoA. Now this time, this is the carbon group that we are losing as carbon dioxide. Now we can see over here that we're also reducing NAD plus to NADH. So that means we must have a dehydrogenase enzyme. And this enzyme is known as alpha ketoglutarate dehydrogenase. So alpha keto dehydrogenase. Now, once we get our succinyl-CoA, now keep in mind that we also had to add in coenzyme A over here. And now coenzyme A was added to this carbon over here. So this carbon, it used to have this COO group, but now it has that coenzyme A group. Now, once we have succinyl-CoA, which is a 4-carbon molecule, so see, we went from C6 to C5 to C4 because we're losing that carbon dioxide. So now we have succinyl-CoA. Now, succinyl-CoA is converted into succinate, in which essentially we are now going to break this bond over here with the coenzyme A, and that release of energy is going to be used to make GTP or ATP. So essentially what happens, we cleave this bond, we are still left with a 4-carbon molecule, but that energy release was used to make GTP. So we see 1 GTP produced. Now we have succinate, which is a 4-carbon molecule. Now, once we go from succinate to fumarate, oh, I forgot to mention, the enzyme catalyzing this step from succinyl-CoA to succinate is succinyl-CoA synthetase. So it's succinyl-CoA synthetase. Now, once we have succinate and we go to fumarate, we see that we're producing FADH2. Now, similar to producing NADH, we know that whenever we're producing NADH, we need a dehydrogenase enzyme, and it's the same case for FADH2. We need another dehydrogenase enzyme. And in this case, it's going to be succinate dehydrogenase. Now, succinate dehydrogenase. Now we see that FADH2 is produced, and so we have to be losing hydrogens and electrons from somewhere, and we're losing them right over here. So if we lose those hydrogens, carbon needs 4 bonds. So in order to fulfill its 4 bonds, it's going to make a double bond with each other, and we see that over here. We see the double bond, and we get fumarate, and we still have a 4-carbon molecule. Now what we're going to do is we're going to hydrate the fumarate molecule, and when we hydrate it, we're adding H2O. When we add in H2O, we get L-malate, and we see that we added in the H2O right over here. Now, it's L-malate because this OH group, it's pointing to the left, so it's levorotary. So this step, going from fumarate to L-malate, it's catalyzed by the enzyme known as fumarase. And once we have our L-malate, we can see that we're going to oxaloacetate, and we're producing NADH in the process, so which means we must be using a dehydrogenase enzyme once again. And in this case, it's going to be malate dehydrogenase. And once we get our oxaloacetate, we've regenerated it, and now this reaction can occur all over again, where we see oxaloacetate combining with acetyl-CoA, forming citrate, and then so on all the way through. The last thing I want to mention is that this process occurs in the mitochondria, but glycolysis, it occurs in the cytoplasm. And that is everything that we need to know about TCA.